get when you cross a Smurf with a cow? Blue cheese!
Blue cheese is cheese which has been inoculated with penicillium mold cultures, which form blue-green streaks, patches or veins. Penicillium roqueforti is a common saprotropic fungi. It is the common penicillium used in blue cheese. It contributes to the appearance and to the pungent aroma. These cheese are aged in a controlled temperature environment like cave.
Starter culture like lactic acid bacteria grown in lab is added to convert lactose to lactic acid. Then, rennet, an enzyme which coagulates milk fat, is added. Cheese curds, which is the solid material which will become cheese, are cut to expel liquid, known as whey. Salt is then added as preservative and flavoring. Cheese are put into mold for specified shape. Whey is further removed by gravity or when it is pressed. The manufactured cheese is then allowed to ripen where flavor and texture characteristic of the variety is developed. Enzymes are released by the dead starter bacteria. At this stage, temperature and humidity at the stage is monitored closely until it reaches optimal ripeness.
In blue cheese, penicillium is added to the cheese at different stages in the making process. This depends on what type of blue cheese is made. For some, it is added to the milk at the start of the process, whereas others are sprayed on to curds before shaping. After introducing the molds into the cheese are pierced with tiny holes to create tiny openings. These tiny holes feed the mold by allowing oxygen to enter. Penicillium roqueforti develops the unique flavor of blue cheese by metabolizing lipid and protein. The metabolizing agents present are lipase and protease enzymes. Penicillium roqueforti possesses two lipases with pH optima at 7.5 and 9.5 respectively. Proteases express two extracellular proteases, which are metalloprotease at optimum pH of 8.0 and aspartic protease. Lipases provide flavorful fatty acids and methylketones by hydrolysing triglycerides in milk. Triglycerides consist one glycerol molecule and three fatty acids molecules bound together by ester bonds. Lipase catalyzes a hydrolysis reaction where water is used to break the ester bonds and to form glycerol and fatty acids. The volatile flavor compounds in blue cheese is N-methylketones, alkane 2,1s, which are catabolized from fatty acids by beta-oxidation with penicillium roqueforti as a biocatalyst. The methylketones can be reduced to secondary alcohols in anaerobic conditions. Types of alcohol are dependent on the strain of penicillium roqueforti used. The production of short amino acids influence the flavors. These amino acids are obtained through the hydrolysis of protein. Proteins are made up of individual amino acids linked by a peptide bond. These caseins are first broken down by enzymes in the rennet. It is then hydrolyzed at an optimum 10 degrees Celsius by the proteases produced by penicillium roqueforti. The hydrolysis of peptide bond is catalyzed by proteases which form amino acids. However, specific characteristic flavors of blue cheese originate from lipolysis and a significant production of methylketone. Penicillium roqueforti is the biocatalyst used in production of cheese. The enzyme in the biocatalyst lowers the high energy of transition state. The microbial byproducts enhance flavor and thus adding value of product. Also, bioprocess is used in manufacturing of cheese because it is virtually impossible to just use chemicals. The penicillium roqueforti mold are harvested from bread. Traditionally, cheesemakers leave loaves of rye bread in the caves. The loaves act as the host to ambient mold in the air. After a month, the mold inside the loaves is dried, ground and combined with cheese curd. This mold cultured on rye bread is not the same as those found on normal loaves.
However, these penicillium roqueforti are cultured in the lab and sterile packed and can be obtained from GN Limited and DuPont. The packed molds powder are mixed with cold boiled water and then sprayed on the dry cheese before ripening commences. Powder can be mixed with milk at renneting stage as well. The problem encountered in producing blue cheese is the time required to develop flavor, uniformity of mold growth and color of the curd. Different engineering approach has been employed, like increasing the temperature. However, it encourages activity of other microorganisms. This problem is still being addressed. Stability of enzymes is also a concern. Enzymes lose their 3D structure and functions beyond their optimum pH and temperature range.
Different sensors are used to monitor these conditions. The environmental problem is the high carbon footprint. High volume of milk is used to produce a pound of cheese. Different approach like heat recovery projects, using renewable energy source and reducing ripening period using adjunct started culture can be done. Penicillium roqueforti has the ability to produce mycotoxins under certain fermentation condition. However, it is not harmful to humans, animals or plants. British blue cheese annual sales amount to £33 million at the end of year 2010. The volume sales of cheese has increased by 1.4% year-on-year as of October 2013, producing about 500,000 tons a year.
Most blue cheeses carry unique names. These names are referred to their protected designation of origin. They bear the names in the region they have been made.